So good day, I am Robin Foà, and I am a Professor of Hematology. In fact, better, I am Emeritus Professor of Hematology at the Sapienza University in Rome. Since I, in fact, retired a few years ago, I'm Emeritus and I continue a lot with research, and also, since it's a topic today, also in the development of clinical trials, particularly in ALL, which indeed is a topic of these video recordings. So the question I've been asked today is relates to clinical trials and what do we mean with clinical trials, the value of clinical trials, what is the benefit, what we get out from clinical trials, benefit for the community, for knowledge, and obviously for patients. So first of all, what clinical trials are? Clinical trials are, putting it broadly, or the way we advance in optimizing management treatment of patients. That's the bottom line. All advancements that we've witnessed over the years derive from clinical trials. No drug or drug combination or will ever be approved by the regulatory agency. These may be FDA in the States, maybe EMA in Europe, and national regulatory agency too, if data don't come out of clinical trials. Data from single institutions are less strong. It's the official clinical trial that lead to the approval of drugs. Why? Because there are certain rules that should be fulfilled or must be fulfilled in clinical trials, and it's the only way we can reach a firm conclusion on the added value of a given drug or drug combination. Now, without entering too many technicalities, the different levels of clinical trials, going from the phase 1 to the phase 2 to the phase 3, 4. Now, the phase 1 are the more experimental, where we're testing really new drugs that have gone through preclinical studies and have gone through many levels of investigation to an extent that the body of evidence that have been accumulated suggests that should enter the clinical arena. So go to the phase 1, which are safety studies to define the safety of a given drug and the maximum or optimal dosing that should be used in patients. If this is all reached, then you go to phase 2, which is a small study to see the benefit of this drug combination. And then the final step is to reach a so-called randomized study, phase 3, 4. Randomized where you're comparing, let's say, the investigational arm with the control arm. The control arm should be the best approved treatment strategy for that given disease. And this is the correct way of behaving, and it's the only way we can really advance in knowledge and management. And nowadays, luckily, the design and activation of clinical trial has become more rapid. So always more drugs are being approved for the benefit of patients. This is something I think it's very important to underline. I said what clinical trials aim at and how trials selected. So, this can be seen in many ways. I have been working in hematology for many decades, and I've been interested particularly in lymphoid malignancies, acute and chronic lymphoid malignancies. Here we're talking of ALL, and ALL has been my major interest for a very long time. And I've worked very actively in a laboratory in the clinic. And we've been designing and running many clinical trials for ALL patients. So what is the benefit? The benefit is what I mentioned, is that we want to improve the management, and the bottom line is that we want to impact on the survival of patients with acute lymphoblastic leukemia. Now, there's been major improvement in ALL over the years or over the decades. Childhood ALL is a typical example where prognosis has changed dramatically over the years. And now in the best centers or in the good and best centers, we can talk about cure rates that are in the range of 85 and 90% of childhood. In ALL, not as good, but I think it's fair to say nowadays that this is improving very much, and the curves are getting better and better, particularly in some subset of ALL. And you have in this website, you have other colleagues who discuss this, the different subsets of ALL and that they have different genetic characteristics, and this translates also in different clinical approach for the disease. So clinical trials have certainly impacted. Now, I've been working actively on this now in Italy, where I've been living and working for most of my life. We have two corporate study groups. One is for children, called IEOC, and one is for adults, called GMAIM. And through the GMAIM, so we've been running many clinical trials in ALL. I've been the coordinator, the PI on more studies so far, and these have certainly impacted. So I would say that there are two big groups of clinical trials. One are clinical trials that are, let's call them investigator-driven, and these are from corporate study groups. And the others are protocols that are designed by pharmaceutical companies. And obviously, the company then approaches the centers of hematology around the world. They identify the centers that respond to certain criteria, and then you put together, I don't know, 20, 30, 50, 100 hematology centers around the world or in Europe or across the Atlantic to fulfill the criteria of the pharma design clinical trial. What we have been, to be very open, more rigid, the one that we've been running, so investigator-driven through the corporate study group. This is what has been happening more frequently in Europe, not only in Italy. Most of the large countries have their own corporate study group. And some studies now are not only national, but European. So this is where we stand. I mean, I've had a major interest. I've been running for the last, 30 plus years, clinical trials for ALL in Italy through the GMAIM. And this has been very important because you're offering the best, the most advanced treatment to a patient. But if a patient is enrolled in a clinical trial, this patient must be assured that he or she is offered the best treatment. In terms, if it's a randomized study, then if you go in the control arm, you're receiving the best approved drug combination. In the experimental arm, you're receiving what you hope will improve on what you obtain in the control arm. So that is a major step forward. If you go into phase 2, you're certainly entering a protocol where all the preclinical studies and the phase 1 studies have indicated that the drug or the combination appears to be highly effective. So extremizing in a way, I always used to say that ideally we would like all patients to be enrolled in a clinical trial because we're offering the best treatment, and I would add also the best management. But if we limit to ALL, but it's the same for other diseases in hematology, if you enter a clinical trial, very often there is a much broader biological workup on this patient. Again, I'll give you an example in ALL. All the studies that we've been running in Italy, they've been nation-based, about 50, 60, 70, or 80 even centers in Italy that participated in a trial. It means that for ALL, all samples of patients enrolled at diagnosis are centrally processed. So the diagnostic workup of each sample is uniformly carried out with certified technologies in certified laboratories. That means that a very precise workup and diagnosis. This translates also during the follow-up of patients. In ALL, it is a key factor that the patients become, as we say, minimal residual disease or measurable residual disease negative. And this is done mostly by molecular biology. If you don't become MRD negative, then the disease is still there. So you have to become MRD negative. Molecular, as I said, and again, at least in Europe, in Italy, and there's also in many other corporate studies in Europe, all the MRD again is done centrally. So it's in recognized laboratory. So the standard is higher than if it was done in every center, and not centrally. So there's an added value in all this. So this has allowed to greatly benefit. I'll give you one practical example. I'm not talking of single diseases, but I'll give you the example of Philadelphia positive ALL. This used to be the hematologic malignancy with the worst clinical outcome, a very unfavorable subgroup of ALL. And I may add that it's the most frequent genetic subgroup of ALL in adults. And the incidence increases with age. So in the over 50, about 1 out of 2 ALLs carry the BCR-ABL, so the genetic marker of Philadelphia positive ALL. Now, the whole story has changed dramatically since many years ago. Tyrosine kinase inhibitors were started to be utilized in Philadelphia positive ALL based on very positive results that we had obtained in chronic myeloid leukemia, a chronic disease that carries the same genetic abnormality. And based on those data, the first TKI, which was imatinib many years ago, was applied to Philadelphia positive ALL, and this completely changed the management of the patients. Now, we as GMAM have been frontline runners in this, and we started, I'm happy to say, 25 years ago. We're celebrating 25 years. In the year 2000, we opened the first clinical trial, which was a phase 2 clinical trial for adult elderly Philadelphia positive ALL patients over the age of 60 with no upper age limit, where we omitted systemic chemotherapy induction, and we gave only a TKI plus steroid, which in those days was imatinib, the only TKI available. Why the elderly? Because it was not easy to consider not giving chemotherapy to what was the most unfavorable subgroup of ALL. The point was that the elderly patient very often could not put up with chemotherapy. Toxicity was too high. So in the real life, most patients received palliative treatment. So indeed, all ethics committees approved, and we opened the protocol, as I said, in the year 2000. This was a very important story because virtually all patients in that protocol, the older one was 89, went into remission. I'm not saying they were cured, they went into remission without chemotherapy. Well, I'll make it a long story short, but that opened the whole story. And over all these years, we conducted trials for patients in Italy where in induction we always omitted chemotherapy. We used the first, second, third generation TKI and steroids. And the result was that the large majority, over 94% of patients, obtained a remission, and many became MRD negative. So this was a very important step forward. Remember that tyrosine kinase inhibitor given orally with a pill, so it makes life much easier, and the toxicity is limited. The next step in this was the addition and consolidation of immunotherapy. Again, you've got some videos on immunotherapy. So we activated the first study, giving one TKI inhibitor, secondary Tazadi, followed by monoclonal antibody BLIA2R. Results were that for all adult patients, irrespective of age, 18 years up, no upper age limit. And this proved, and was published in the New England Journal, that you can control the worst hematologic malignancy with a targeted therapy followed by immunotherapy. There has been a major change, and now we just completed another study with third generation TKI, and this has been the first phase 3 study. So there's a randomized control arm with a TKI plus bleed and experimental arm. So this is a major change. We would never have reached these results if we hadn't activated clinical trials to do this, because without that, this would not have been possible. And this is one example of how we are totally revolutionizing the management of the most lethal leukemia. I can also add that in phase 2 study, so-called the ALB with Tazadi and Blina, the long-term follow-up of the study showed that 50% of the patients received only a TKI and blinatumomab. They never received chemo, and they never went to an allogeneic transplant. So a major shift forward. So I think this clearly makes it evident how a participation in clinical trial is an added value to patients. And we would like, as I already said, that all patients in the trial, we would like to design protocols as we would like, because I think the most independent one is investigator-driven. This in real life is not always easy, but we have to make all efforts to do the studies that we feel are optimal for patients. And that is really what I think we should say about this. Clearly, you can adhere as a center and therefore the patients to or manager gives, and also on pharmaceutical studies. I don't want to diminish this by any means. We've done, when I was head of hematology in Rome, I mean, we had an enormous number of clinical trials open in all hematological malignancies. So we took part in many, many trials, phase 1, 2, and phase 3. And that was extremely important because we could, if you're doing phase 1 trials, you can test innovative drugs. Then it's very important that in parallel to that, you have the strength to run your own studies to our corporate study group. So I think it's a combination benefit. And as I said, this is for patients and it's for the advancement of knowledge. And if in hematology, macrophages change dramatically over the years, this is largely, or I would say 100% merit of the clinical trials. And saying this, I just finished by remembering that this has led to treat many hematological malignancies without chemotherapy. I mentioned chronic myeloid leukemia before. This was a lethal condition, and now with TKI, this has changed completely. I just wish quickly to remember acute promyelocytic leukemia, a difficult to manage condition. Now most patients are managed without chemotherapy. I mentioned Philadelphia positive ALL. I could continue. I mean, many lymphomas are managed without chemotherapy. Hairy cell leukemia is changing, and probably in the future, again, chemotherapy may be omitted. So this is a major change following the advancement of knowledge and the design of optimal clinical trials. So I'll stop here, and I thank you very much for your attention.